Okay, thank you all for coming on this rainy day. And today it's my great pleasure to welcome Randy Nesse, who doesn't really require that much of an introduction. He is our founding director of the Center for Evolution in Medicine and has just stepped down from that large task.
Right.
And he's going to tell us what he's been up to as of late with inherently vulnerable biological systems, how maximizing genome fitness causes phenotypic fragility. Thank you.
Thanks, Ann. So it's a great chance to talk with you all. I'm really glad to have this opportunity. I should say that I'm really hoping the purpose of this talk is really to illustrate what I envision CEM doing, which is creating interdisciplinary opportunities for people in different fields to get good advice from each other. And I'll emphasize I'm not a population geneticist, and my talk is not really about population genetics, but I think it will inspire some geneticists to come up with some pretty interesting ideas about why highly heritable diseases persist. So we begin, however, with intrinsic vulnerabilities. I'm a Detroit kid, and I used to fool with engines. I still fool with engines, although they're getting harder to work on these days. There's some things that engineers have a devil of a time fixing. If you've got a car with a carburetor, it's prone to flooding. If there's too much gas in here, it won't start. Or knock. You know about knock. You have to buy expensive gasoline to stop your car from knocking. It's because the timing of compressing the air and the gas has to be just exactly right. Otherwise, it explodes in an irregular way. Backfiring, likewise. And then there's vapor lock, which is a chronic problem for any car that's carbureted, because if the fuel pump is trying to pump gas, it can't do it. Again, talking with engineers in Detroit, these are not fixable problems. You can minimize them if you're willing to push the trade-offs way towards the point of engines not working very well, but you don't want that. And so if you have an old-fashioned gas engine that is not fuel injected, you are going to get these problems. Why is this important to us? Let's look next at the Airbus. You all have been reading in the papers about it. So the simple explanation is the plain old medicine one. Yeah, the plane wasn't quite right, and the pilots weren't trained right. That's the simplistic explanation. An evolutionary approach to that kind of thing is the approach of an engineer. An engineer asks, why was it designed the way it was? First thing was there was big-time competition between two companies. Boeing people had to get out their new plane fast, otherwise they would never sell another plane, because Airbus had something more efficient. What did they have to do to make their plane work? They had to move the engines. They had to put on new engines, which were too big to fit under the wing. So in order to do that, they had to move the wing forward. When they moved the wing forward, the plane had a prone tendency to stall and fall out of the air. To fix that, they added a sensor at the front to say when it was about to stall, and automatically, when it was about to stall, turn the nose down so that it wouldn't fall out of the air. Except that if that sensor failed, then it thought the plane was stalling, and it pointed the nose down. And if the pilot didn't know how to turn off the automatic device, the plane dove right into the ground, and that happened twice. And some of you who are planning holiday travel won't be able to get on your planes because all of these things are still grounded. The big thing here is that the design for this plane was optimized by very smart people. There were no errors in the blueprints equivalent to genetics being normal. The construction of the plane was perfect. There were no construction abnormalities. It was operated within the bounds of normal range. Essentially, it was intrinsically vulnerable because of trade-offs that pushed the design of this plane close to a cliff edge where failure was likely. Now we're on to medicine and why diseases exist. The thing I was always taught is you got to figure out the mechanism and what's broken. Individual differences from genes, environment, and development explain why some people get sick and other people don't. That's medical research. Those are proximate explanations, as you're probably all aware. Those of us in the Center for Evolution Medicine are looking for evolutionary explanations, and there are two very distinct kinds. One is phylogeny, Darwin's #1 discovery about the unifying of all life, and many geneticists are mainly interested in the relationships between organisms and how you can trace those relationships. That's there. The other part is adaptive significance. That's what I've always been interested in, is why aren't traits better? And this has led to a lot of interest in trying to figure out which of these 6 possible explanations for why traits aren't better explains one particular thing or another. Constraints, mutations happen. Living in modern environments is the main thing that's responsible for cardiac disease and autoimmune disease. Pathogens evolve faster than we do. This is the core of evolutionary medicine. But now I want to address a different problem, related. Why do alleles that harm fitness persist? We're going to go to the genetic level and try to ask why they persist. And the standard answer, of course, is, hey, they're selections and drift. And then natural selection can't keep up. Also, changed environments make a lot of previously neutral alleles negative, and so they're only gradually selected out. Some of those are mismatched, others extend the lifespan in modern environments so that things that weren't diseases before now become diseases. And finally, there's balancing selection, vastly overrated, but of great interest to a lot of people. So the usual model for explaining these things is to assume that alleles associated with disease are deleterious and gradually being selected out. And second of all, to assume that maximizing fitness will minimize disease vulnerability. There's our model. You've all seen that in your textbooks 100 times, right? Natural selection pushes the trait, squeezes it together, mutations squeeze it out, and that's, whoops. So what's missing here, though, and the whole point of my talk today is to really encourage us to not just think about the trait distribution, but to think about the fitness function. How is fitness influenced if the trait is at different levels? Here it's maximized, lower on the sides. With this trait distribution, individuals out at the tails are prone to disease. No surprises. And then, of course, if the fitness function is squeezed or the trait distribution is widened, you're liable to have more disease because there's more overlap here. All very straightforward. My thesis today, though, is that this may not be adequate. These are obviously important, but do they provide a full explanation for what we're observing? Point number 1, selection stabilizes traits at values that maximize genome fitness, often at the expense of health. 2, variation in such traits will lead a few individuals to be off the cliff and vulnerable to disease. And 3, this may help explain what we've been finding with all of our GWAS studies, which is high heritability from tiny effects of many, many nearly neutral alleles. Put another way, don't just assume that disease genes are abnormal and being selected against. They may be normal genes that benefit their own transmission at a cost to health, and they may be stabilizing a trait at a point on a fitness landscape that consigns a few individuals to disease. So I got into this because of concern about schizophrenia. If you treat people with schizophrenia, it takes you about 2 weeks to realize, gosh, 1% of the whole population has this devastating disease. And it cuts their highly heritable, and it cuts their fitness by half. You've got to ask yourself, why the heck does this persist? Some people say benefits to kin of creativity or something. There's no evidence for that. Furthermore, there are no common alleles with large effect. And few alleles with large effects, few rare alleles. And oh boy, if you go talking about this with psychiatrists and others, you get all these wacky ideas. You get the idea that, my students keep saying, oh, what's the adaptive advantage of schizophrenia? There must be one. This logic of because a disease is highly heritable and devastates fitness, it must be useful somehow. That is the biggest mistake, but a very common one in evolutionary medicine. So I actually got into this because of an article in Behavior and Brain Science by Burns in 2004. I was asked to comment on it. His idea was that because schizophrenia was 1% in all populations worldwide, it must have to do with some gene that was with us before we came out of Africa, since it was uniform across the globe. And he thought probably it was hitchhiking. There was some useful gene that got selected for and it dragged along deleterious genes. So I thought about that for a while. It's been a long time since we left Africa. And even if you're not a population geneticist, you can calculate how many episodes of recombination it takes to separate that kind of thing so that the hitchhiking fails. And it's not going to work. That's not a potential explanation. So I spent a month just thinking about it. And then I read David Lack's work. David Lack, of course, is a famous ornithologist. And one of his wonderful experiments was asking, how many eggs should a bird lay? Why not lay an extra one and have an extra offspring? And he did the experiment of taking eggs and moving them from one nest to another, and discovered that if you added an egg, you got like half an extra offspring. If you added 2 eggs, the whole nest might fail. The key inspiration for me was David Lack's observation that this fitness landscape was asymmetrical, and pushing it up one stage too far made the whole thing crash. Then I started wondering about horses. Did you know that out of every 1,000 starts in a horse race, the horse is going to break its leg? That's why they've always got somebody with a gun, or at least a horse trailer, standing behind. That's tragic. Why do horses break their legs so often? It's because they're bred for speed. Only these horses get bred way up here, and they have longer, thinner, lighter cannon bones, which are more prone to breaking. The importance of this for schizophrenia is that everybody is selected for speed very intensely. And this means that those who break bones are not much faster than others, and their relatives aren't much faster than others. They're all very, very fast. They're all pushed right up to a peak where they're liable to break. And if you think about it a little bit more, you realize that because there's so much variation around this peak, natural selection pushes the trait back down that slope just a little bit. More about that later. Next, I'm going to provide you with a very quick overview of schizophrenia genetics. This could be for diabetes or autism or others. I've chosen schizophrenia because we have more data on that than anything else. When I started doing this, there were all kinds of ideas out there about schizophrenia. And in just the last 18 months, there have been 20 papers that open up this whole area and reveal what we're dealing with. Very highly heritable, 0.8 or so depending. In 2000, we thought we'd find the abnormal genes. Labs around the world went to work. A whole bunch of people who wanted to go into evolutionary medicine came to me and said, I want to do evolutionary medicine. I said, no, you want a job. You should go into genetics. And some of them did. Many candidate genes were identified. This is what we expected to find. We expected to find some genes that would explain vulnerability to this terrible disorder. What we actually found was more like this. Jackson Pollock to the max. It's a mess. 25 candidate genes were observed, and people were very confident. And I should emphasize, people spent their whole careers trying to find these genes. And they spent billions of dollars over a decade or more. So 25 candidate genes were identified. My former student, Matt Keller, with a group of other geneticists, published this 3 years ago. Not one of those candidate genes panned out when put in a larger database. Every single one was a false positive. Jeez. The same group also did this graph showing that the very common alleles had minuscule effects on fitness, maybe increasing your risk by 1% or 2. The only ones with any substantial risk increase in fitness were copy number variations, which are very rare. But what they then calculated, which I think is fascinating, is that all of these are on a line that explains the same proportion of variance, 0.04%. That's not very much. And we have to ask, why aren't there any alleles out here that are both common and substantial? A terribly important finding, not just for schizophrenia, but for almost all complex genetic diseases. So here's the latest from the giant working group of where we find documented alleles that do influence schizophrenia. There are 104 of them illustrated there. Do you notice where they're located on the genome? Which chromosome they're on? Yeah, there's a lot of DHLA locus on chromosome 6. But the big point here is that they're equally distributed over the entire genome. Furthermore, a study that just came out finds that their background selection is quite powerful on these alleles, but there's no evidence of positive selection on these alleles. And this also is just out and very cool. This looks at what kind of genes are the ones that have allelic variations that influence the risk of schizophrenia. The red bar is the no enrichment. The big blue one is loss of function intolerant CNS-related genes. The black one is other loss of function intolerant genes. And then there's the rest of them. So something substantive is going on here. And this one is just astounding. Everybody thought, oh, if we can only sequence the whole exome, we'll find it because we know that genes have they're influenced by proteins. Look, only 1.1% of these loci are expressed in exons. All the rest are in other regions. Is this getting complicated enough for you, or pessimistic enough for you? Let's take one step further. Now we're doing gene correlations to find out whether specific genes go to specific disorders. And the answer is, our entire idea that there were going to be specific genetic abnormalities causing specific diseases with specific pathology is false. And we know it's false now. Look at this. Here's the correlation between major depression and anxiety disorders. They're basically the same genes. Very highly correlated. The other big one is bipolar disorder and schizophrenia. That correlation is something like 0.7. Which alleles influence these disorders? There's massive overlap with all of these. People have even talked about a G factor that influences mental disorders in general. It seems like the way we've been thinking about these disorders is not working. Here's one that did work. This is a group that looked at shared bipolar schizophrenia alleles, those a few that I talked to you about before a minute ago. And it turns out that 3 of them are in the synapse, they're expressed in the synapse for long-term potentiation. That's very cool. This is what people keep trying to do. They try to find loci in specific genes that have specific mechanisms. So that's cool. But that's about the only thing that's specific that's come out of this. So here's a quick summary about schizophrenia. 1% worldwide, really high heritability, really low fitness, and thousands of alleles all across the genome, spread so that the number of alleles on any chromosome is directly proportional to the size of the chromosome. Background selection is prominent, no positive selection, non-specific effects on many disorders, and if you do a polygenic risk factor, which was hope, you take all the variations, even if they don't reach significance, and put them into one algorithm. It works to predict risk of schizophrenia in Europeans, and then you try it in Africans, it doesn't work anymore. Jeez. It's worse than a Jackson Pollock. Let's do autism for a second. Maybe that's better. 1% prevalence, recurrence rate of 20%, heritability about 0.7. We know that only about 20% of the de novo variants, or only 20% of the variations are de novo variants, 50% from additive tiny effects from hundreds of genes, and none of which are present in more than 2% of patients. What? We're looking for what combination of genes, and they're not even present. And there's massive comorbidity for autism with intellectual disability and epilepsy. This is the newest results about what kinds of genes are involved in autism. Here, in the pink are ones that are identified. This is non-additive. This is predicted common inheritance. That is, 50% of it is small alleles with minuscule effects, and another 41% is not accounted for. Very much the same picture as we saw for schizophrenia. Epilepsy, again, heritability about 0.8, devastating to fitness. Recurrence risk is 7-fold. If your sibling has it, your chance of having it is 7 times greater. About 25% from common variants that are identified, maybe as great as 50% if you expand it. They had a bunch of candidate genes, and none of them were panning out once you take a larger sample. There are few rare variants in each of 1% of patients, and they make a big deal out of that because they are in loci that influence polarization of neurons. But they don't account for but a fraction of the variants. Interesting risk factors, trauma and drugs and fever and infection and stress can all make epilepsy more likely. Wait, it's genetic. Wait, it's environmental? It's environment acting on some system that I would say is pushed very close to a cliff edge, where it's vulnerable to failure. Bipolar disorder, 1% prevalence, you get the pattern here. Heritability very high. And this summary of this, the most recent, is out of 22 large GWAS studies, only 2 found a significant locus in discovery and replication stages, 7 found none. That's disappointing. Again, whole careers spent on the assumption that specific disorders are caused by specific genetic abnormalities that we can find that will lead us to pathophysiology, will lead us to cures, and it's not working. Furthermore, bipolar has this big overlap with schizophrenia and autism. So what are we going to do now? What we're doing now is larger and larger GWAS, trying to find genes of ever smaller effects, and that's useful and interesting. But the big picture remains very pessimistic here in terms of our hope back 15 years ago that we would find genes that would explain these diseases. So here's a thought experiment I'd like you to go through with me. What if we could just eliminate all mutations and eliminate environmental change and eliminate developmental stochasticity and let things run for a few hundred generations? Would this reduce disease vulnerability? Sure it would. It would a lot. But completely? My answer is disease would persist. And the reason I think it would persist is because selection maximizes genotype fitness at the expense of increased disease in some individuals. I'm going to give you two kinds of examples. There are more. First, maximizing genotype fitness can cause phenotype vulnerability. And two, traits with cliff edge fitness functions are stabilized at values that leave some individuals vulnerable. And these are the list I'm going to go through very quickly. I mean, each of these, sexual selection, antagonist, all of these harm the health of the individual, at least at some point, but nonetheless are selected for. Cui bono is a phrase lawyers use, and George Williams and I asked each other that often about diseases. Who benefits? Who gets the genetic benefit from these things? Again, the big picture here is the fitness function and the function that maximizes health are not necessarily the same. It might be that your health function is here, and given that fitness is going to be maximized here, your health is decreased by 50%. This is a project that Dan Kruger and I did years ago, looking at the male mortality rates by age by 20 different countries. And the big picture here is that they're 3 times higher for males in almost every country at the onset of sexual reproduction. So cui bono, who benefits from this? Genes. In fact, if you think about it for a moment, the whole sex system, ranging from desire to doing things that get you in trouble to taking, all these things, they're good for your genes, and they cause a whole lot of trouble. Antagonistic pleiotropy, the thing that originally got me into evolution medicine, who benefits from these changes, these genes that give you benefits in youth, but cost later in life? In fact, if senescence could be gotten rid of, 1/3 of us would live to be 1,000. It's the genes that benefit. Natural selection could shift this to give you a longer lifespan, but it hasn't done it. Kin selection, that's another example of doing things that harm the individual's fitness at a cost to your fitness. Then there's another category I just started thinking about. I think there may be traits that give benefits to other lucky individuals. Maybe in another generation. It might be that all of us have a little extra body weight we carry around in part because intermittently famines happen, and if they kill off 90% of the population, those individuals who are extra heavy now are going to be success. And then there's winners in winner-take-all contests. If you have a mating contest or any other contest where one individual is going to take all the rewards, there's a whole lot of losers who are going to have health costs from that. But the one individual who wins, that genotype is going to carry on. Then there's sexually antagonistic selection. Sometimes benefits go to the opposite sex at a cost to one sex. In humans, that's hemochromatosis. Women losing a little blood every month means that they benefit from absorbing extra iron. Men do not lose a little iron every month, and therefore, if they have this genotype, they get hardening of the liver, and they're very sick. Same thing with Drosophila. 8% of all Drosophila genes are now known to be sexually dimorphic, sexually antagonistic. David Haig's work on competition between parental genomes, the interest of the maternal genome and the paternal genome are different. And as a result, there's quite a contest over the size of the offspring, and imprinting mediates that possible disadvantages to health. And the last one I'll give you is just plain old parasitic genes that reproduce themselves at the cost to the host. This is the new WTF40, amusingly named. And this is an example of it, actually. The blue is the toxin which kills off all other gametes that don't have the antidote, which is the purple. A very effective way at advancing its own interests at the expense of the host. So what are the implications for complex genetic disease of natural selection shaping genotype fitness instead of individual fitness? Variations that harm health can be neutral, or they can be deleterious but still give a net benefit to themselves. And you wouldn't expect that they can be eliminated by natural selection. It may help explain the persistence of many alleles with tiny effects that cause complex genetic diseases. Okay, on to part two. Part two is about the shape of fitness landscapes. Steep functions with narrow distribution, that's obvious. We'll give you one example. I'll talk a lot about cliff edge fitness functions, which is what inspired me to get into this whole area in the first place. And then I'll talk a tiny bit about how selection for robustness leads to vulnerability, and how positive feedback-prone systems are especially prone to collapse. So this is a steep fitness function. You know, the fitness function and trait distribution, it's really bad. If you have something that's, whoops, there's supposed to be something that says disease there. But that kind of a very wide trait distribution relative to fitness function means that quite a lot of individuals are going to be out at the tails and really vulnerable to disease. What causes that kind of a fitness function relative? Well, arms races in particular, because you get pushed from this side by the host and this side by the pathogen, and both of them are quite potent pushes inwards for the fitness landscape. But this is mainly what we're going to talk about is cliff edge fitness functions. You could call them asymmetric, but I use the frame cliff edge because it's kind of more interesting. Your fitness goes up and peaks here and then falls off a cliff. Your maximum phenotype fitness is, of course, at that peak. Maximum number of offspring. Clear enough? Problem with that, though, is that that's the trait distribution. And if that's the trait distribution, what about these poor folks out here? They're really prone to disease because they're off the cliff. Their fitness values are very low. Maximum genotype fitness is down here. And a lot of these genes are going to be wiped out because these individuals don't do very well. Maximum genotype fitness is pushed down on the shallow side of the slope. There are some individuals who have problems because they have low fitness, but they're relatively few compared with the peak. The third spot is where health is maximized. You can push things further down this slope to a point where you have the minimum proportion of the population with less than 50% fitness. That's my arbitrary definition of vulnerability, and the converse of that is the maximum number of people who are healthy. So the usual focus of people who have worked on this model is to point out that the natural selection moves the mean for a trait value down this slope to an area that is not optimal. This is what's inspired people. There are three pretty good mathematical models of this, and they all focus on the fact that natural selection stabilizes the trait at a level that is quite a ways below what would maximize individual reproductive success. But there's only one other person who's taken the approach that I have, which is emphasizing these poor folks off the cliff here. My question is, how does percentage off the cliff vary as a function of the trait standard deviation and the cliff steepness? I said there was one group that's worked on this. Mihaela Palichev gave a talk in our series last year. I'm not sure how many of you remember it. That project talked about the birth canal and how large the baby's head could be. And the idea was that a baby's head got benefits as it was bigger and bigger and bigger until it was 1 centimeter too big and both the baby and the mother died. That's a pretty catastrophic outcome. And so their calculations were done on that kind of a cliff edge function. So I needed a model to work on this, and in order to create a simple one, I just made something moderately arbitrary, but I think intuitively attractive. First of all, the fitness function is a normal distribution with a mean of 5 and a standard deviation to the left side of 1.0. To the right side, the standard deviation can vary to make cliffs of different steepness, and the trait standard deviation can be variable to create different spreads. And then the model has to calculate what's the trait mean that maximizes population fitness, that's 5.0. What's the trait mean that maximizes genotype fitness, that needs to be calculated. And what's the trait mean that minimizes the percentage who are off the cliff. And this is the illustration I'll spend just a minute on. So the black line is your fitness function. The blue curve is the distribution of individuals who have a trait mean at the peak, 5.0. We're calling their fitness 1.0 in the first generation. The red distribution is that of individuals at the point on the landscape that maximizes genotype fitness. And the green one is the one that maximizes health or minimizes a percentage who have less than 50%. And here's where the payoff is. What percentage are off the cliff? Well, of those who are at the peak, 23% are off the cliff with very low fitness. 6% are off the cliff from the peak that maximizes genotype fitness, and only 1% are off the cliff, 1% on either side for those where these things maximizes health. And there's quite a lot of cranking of spreadsheets to get all these results, but they're easy to come by. So then I went through and looked at how the percentage who fall off the cliff varies as a function of the steepness of cliff, and the standard deviation of the trait. So obviously, as the cliff gets steeper, there's more and more individuals who are going to fall off the cliff. And furthermore, as the standard deviation of the trait gets larger and it spreads the distribution, more and more people will be off the cliff. Here's 1% off, 2% off, 3% off. The punchline here is that the things we see with the complex diseases that I told you about a little bit ago fit this very well. 1 or 2 or 3% of individuals are, have their fitness devastated by these disorders, which is exactly what you expect with realistic estimates of a standard deviation for the trait and a steepness of a cliff. Reviewers have said, okay, this is fine, but you haven't shown us that selection actually does stabilize the trait at that level. And so I went through quite a lot of trouble to do a sequential analysis showing that if you start at the fitness maximum and look at the trait density times the distribution for each generation, it takes about 10 generations to go from 5.0 down to 4.7. If you start at the health maximum, it takes about 15 generations to go almost all the way to that maximum as well. And my initial intuition that the optimum for the F1 generation, which would be 4.7, actually is the point where it's stabilized, whether you start from the top or you start from the bottom. The point here, again, is that natural selection stabilizes the trait at a value that leaves a few percent of individuals with very low fitness. So now we do mutations on the fitness cliff. And this is the place where I'd love to talk with some of you who are professional geneticists to get your take on it. It seems to me that, yes, natural selection is going to stabilize the trait here, but there are always going to be mutations going around there. What happens if there's a mutation that pushes it back up this direction? Well, in the first generation, that trait, that gene is going to get a big advantage because there's going to be a big payoff from pushing up that cliff in the first generation. Subsequent generations, the next 10 generations, it's going to come back down again. But it seems to me this leads to cycling of neutral mutations. And they're going to look neutral from any of your population genetic models because on the one hand they're getting an advantage here and a disadvantage here, and they're cycling, going back and forth around there. I think that this will create high heritability for disease risk from many neutral alleles. But I think this needs work from other people. I should mention at this point that I really appreciated the chance to talk with many of you here about these ideas to encourage me to keep working on it. There are several people who have been especially encouraging elsewhere. Steve Frank and Carl Bergstrom have been very good. Alan Rogers has also been very helpful. And Fred Niehout are people who are actually know their genetics and have encouraged me to keep pursuing this. The next question we have is, what shapes cliff edge fitness functions? So why the heck do you get one of these really steep things? Well, host pathogen arms races we already talked about. And then there are traits that fail catastrophically. If it's just, a smooth curve, what the heck? No big deal. But if the bone breaks, it breaks. And you're dead if you're a horse. Then there are traits susceptible to positive feedback. I mean, for epilepsy, for instance, the more hyperpolarized you are, the finer it is, the faster you can transmit those signals in your neurons until you get a breakdown that causes a positive feedback, in which case the whole thing goes haywire. And any trait whose values increase exponentially, that's going to push things up faster towards that peak. Cancer, I think, is probably a very good example. How many stem cells do you want to have in a crypt in your bowel? Well, if you don't have enough, they're going to die and be damaged, and you're going to have faster aging of your bowel mucosa. But if you have too many, each one of them increases your risk of cancer. So you get this steady increase of benefits from repairing things, but the risk of cancer increases dramatically once you get more cells. And this might be a very good example. There's a group at Yale that's working on this exact model and finding support for it. Back to schizophrenia. So what the heck is the trait? This is the problem with this whole approach. Essentially I'm saying we shouldn't just be looking for bad genes. We should be looking for traits that have fitness functions that make the system vulnerable to failure. But how do we find those? And do they even exist? This is the big challenge. I don't pretend to gloss it over. So we have to make up stories and try to guess at something we can go looking for. One good guess for schizophrenia is optimal pruning. At adolescence, you lose 20 to 40% of your neurons through a normal process of pruning, which interestingly is immunologically mediated, explaining why the HLA system is so potent in there. But on a much higher level, we all get benefits from creating causal models of the world. We try to figure out how things work, and we say, this is accounted for by that. That is a remarkable capability that our minds have. This is kind of the key to humans having selective advantages big time. If you don't do that very much, if you're a skeptic and you don't think anything is really connected to anything else, you're not going to benefit from this. But what if you are really prone to create stories about almost everything? That guy, I saw Ben was flipping his finger on his lips like that. I think that means that he's having hesitations about. That's the kind of thing a schizophrenic does. They attribute meaning to everything. I think there might well be selection to put that tendency to create meaning right up there towards that edge, and which also explains why so many people are prone to conspiracy theories. Our human minds are pushed way towards an edge. Epilepsy, likewise, this is, I think, the best place to put effort in, because you can actually measure polarization gradients and other things that might influence the risk of epilepsy. The epileptologists are very keen on finding specific genes. And all of the articles about epilepsy genetics I found list genes that do or might have significant association with different kinds of epilepsy. And none of them mention the effect size. They don't point out that most of these have minuscule effect sizes. They're really hopeful that they can find something specific to put their hat on. More polarization you get, this is talking in general terms, the faster you get neuronal transmission, at the risk of the whole system going into a positive feedback loop and failing. Autism, likewise. The kids with autism have brains that are big at birth. They're too big. They grow fast. Is there something about rapid growth of brain or expansion of brain that gives you selective advantages at the risk of connections not developing correctly? This is just wild speculation, but it's the kind of idea that you come to when you're trying to figure out what might those alleles be doing that would push a system up towards an edge where things fail. So the conclusions here are pretty simple. Natural selection maximizes genome fitness despite recurring and resulting health risks for some individuals. And this might help to explain highly heritable diseases, caused by the tiny effects of many, many nearly neutral alleles. For next steps, there's just so much to do here. What kind of genetic signals would reflect cliff edge fitness functions? I mean, is there some way we can look at that place I talked about cycling between the effects of mutations pushing up and pushing back down, where we could look for those effects? You know, I can imagine, for instance, looking for associations amongst effects of alleles to cluster them in two different groups, one of which pushes a trait this way and the other which pushes a trait that way, to see if that might reveal something. How can we look for traits with risky trade-offs? What kind of trade-offs are we looking for? This whole area is not going to go very far until someone can find a specific good example where it helps to promote things. And what diseases will be best to study? I mean, I've given you a few examples that I think are interesting, but I think that's another project. And many of you may be working on other diseases that are relevant. And it might be that other people who are watching from afar on the live stream are having other ideas. I'd like to hear from them as well about their responses to all of this and their suggestions for how this could all go further. A lot of what I've talked about is in chapter 14 of my new book. That's kind of a layperson's level of this, and I'll be working more on this, and I look forward to talking with lots of you about how it can move forward. Thank you very much. I think we have plenty of time for questions, and a microphone will pass around. I'm not sure who can help pass the microphone around.
Very good talk, sir. Just one question I have is, what do we do with this information when it comes to treating or curing these sorts of things? Is gene editing still viable, or do we need to look at some other avenues?
So the great hope has been that we would find specific allelic abnormalities and be able to do something about them. But it's really clear there are so many of them with such tiny effects that that is not going to work. We need a different approach. Yeah.
One quick question. It was unclear to me how the recurrent mutation, when you were saying this recurrent mutation on the slope would be contributing to high heritability. So, because one doesn't normally think of heritability as being tied to high mutation rate.
Yeah, I'm not sure it would increase the rate of heritability above what was already there, unless it actually increased the amount of genetic variation that was persisting, because of the fact that these mutations that push things up that direction, instead of being wiped out very quickly, which they would otherwise, they might persist for that 10 generations or so. And so I think it would keep these mutations that influence the position of this trait, they would stick around longer as a result of that. Did it answer it, do you think? Or?
Can I add to that explanation?
Only when you get the microphone, Manfred.
This is a very intriguing theory, one of the best I've heard in a long time. But it leads to questions of therapies, potential therapies. If it's not a genetic problem, then obviously everybody's looking in the wrong place. But if it's a developmental problem, that is, it's problem with brain development in adolescence, then what would be the proper research strategies and therapies to address your claim?
So, good point. You know, one of the things that's gotten more clear to me as a product of this work, I gained new respect for developmental stochasticity. I mean, any system that's in development is going to have neurons wandering different places and different kinds of imprinted genes showing up in different places. Now, natural selection is pretty strong at tamping those down and canalizing things so that the phenotype ends up fairly well controlled. So all of these disorders are developmental disorders in that developmental variation, both from genetic vulnerability to variation and environmental perturbations. But I think your point, I mean, again, if you get the influenza during pregnancy in the second trimester, you're more likely to have a child who has schizophrenia. That people have talked about that, and especially toxoplasmosis also. There are a lot of things that influence brain development and people talk about it as if there's environment or genes. It seems much more likely that the whole system is shaped to put that system again close to a cliff edge, such that those who are genetically vulnerable are more vulnerable to getting their epilepsy, their autism, or schizophrenia as a result of any kind of thing that interrupts or disrupts brain development. So I think this helps us to stop thinking about genes or environment and see how the genetic predisposition essentially makes the system more vulnerable to perturbation in the process of development. Now, Manfred.
So there is a formal reason why you would expect to have high heritability once you are up there on your peak, because it basically represents an equilibrium point for a population. And if even if you have alleles that have high degrees of nonlinearity in their effects, once the population is near an equilibrium point, the additive component of those alleles dominates. So you have a higher increase of the additive genetic variance up there, which translates into higher heritability.
Interesting. Thank you. Jim.
Nice talk, Randy. Thank you. Have you thought about making the x-axis, the trait value, multidimensional?
I knew someone would bring that up. The first thing wrong with this whole thing is, are there really trade-offs that leave traits to a cliff edge? Or is the whole system integrated so thoroughly that it doesn't really make sense? I don't know the answer to that, but I think simply conversing about that could help hone that in. Part of the thesis here is that it might be that these cliffs are special kind of things because of a special kind of trade-off between just one or two or three variables that are pushed, just like the gas engine we talked about before. But the other huge problem with all of this is that this all works in n-dimensional space. And you can't even assume that these fitness functions are smooth. They might have wormholes in them, such that everything drops out at one certain point. And it might be that certain combinations of genes and alleles lead to particular vulnerabilities. And on top of all of that, the fact that the predictive risk scores for one population don't carry over to another population. What do we do with that? It really suggests that there are no such things as specific alleles that predispose to schizophrenia in general. For epilepsy, it's a better picture. Epilepsy, the risk alleles that predict in one continent also predict in another continent. But the percentage of allelic the frequency of those alleles is vastly different in different subpopulations of humans, and so that screws it up hugely. But the two big problems with all this, and again, there's always a good reason why something hasn't been done, right? The first one is, how do you actually talk about and look for these kinds of cliff edge trade-offs? And do they really exist? And second of all, the whole damn thing is in n dimensions, meaning that this is a gross simplification. What's the answer to your own question, Jim? What do you think? Could it be modeled in n dimensions and still keep some coherence?
Well, I don't know, but the striking observation is the fact that the prediction in Africa doesn't work in Europe.
Yeah.
Meaning there is quite a bit of context dependence somewhere in the system, whether it's at the genetic level or it's at the strong environmental input, as was suggested earlier on. And what that's going to look like, I don't know. And but at least I think it would be really helpful in theory to at least give some thought to what a multidimensional model would at least need to capture or begin to encompass.
Good. Other questions from anybody? Or suggestions about what disease to look at and how to try to find some way of finding models that can identify signals of this kind of thing happening in the genome.
Quick question with respect to when you were breaking down the phenotypes, did you have, when people have done the genome-wide association studies and rather than using something like schizophrenia, but instead use something like pruning or something like that, do they get different or better, or how do the results look if they use some alternative characterization other than these grab bag larger characterizations.
Yeah, I don't know of anybody who's tried to find ways, Ken, of connecting genome variation with pruning results. But that's a natural place to go with this, and I think people are trying to do that. And I should emphasize to you, I mean, I couldn't have given this talk 2 years ago. The data are just barely out now. And it's a combination of large new data sets and new statistical methods that makes it possible to realize that most of our old ideas about this just do not work. And we need to try to figure out some other way to go forward.
Any other questions? Okay, with
I think that wraps it up.
Thank you, sir.
Thank you very much.